Hello everyone, I'm Sasmitha Dasanayaka and I will be presenting our research on interpretable machine learning for brain tumor analysis using MRI. This research was conducted by Sanju Anupa, Vimuchanta, Dr. Tanuja Begoda, Dr. Dulani Medenia, and myself. First of all, let's have a brief introduction to the brain tumor medical domain. Brain tumors are potentially fatal growth of cells in the central nervous system that cause of the most number of deaths out of all the central nervous system related cancers. Still, medical experts have not been able to discover the exact causes or unique symptoms related to brain tumors. Inability to diagnose and identify their types at early stages further reduces the survival rate of brain tumor patients considerably. With the huge advancement of AI and deep learning in the field of medical imaging, image processing techniques such as image vision, classification, and segmentation help doctors to diagnose diseases as early as possible compared to manual inspections. Now I will state the problem we have addressed through this research. There are many solutions based on deep learning techniques that are capable of classifying MRIs. Deep learning models are inherently complex and less understandable to humans. In other words, those models are black boxes in nature. Existing systems in this domain are focused only on improving accuracy regardless of their black box nature. When it comes to a high-stake domain like medical diagnosis, prediction accuracy is not everything. Understandability and transparency in the decision-making process is critical to apply existing models into the real world. Therefore, the system has to make sense at least for domain experts. As in this image, in the medical domain, there are many number of deep learning models, but most of them are black boxes in nature. In return, they are less human understandable. That being the case for medical practitioners not to have faith in deep learning models. To address the above identified problem, we try to introduce interpretability into deep learning models to make models human understandable. This is the contribution of our paper. These literatures influenced the background of our research. Proposed models, concepts, and results of above three literatures were used to implement classification module, segmentation module, and introduce interpretability to classification module respectively. Here you can see some of the state-of-art work on classifying brain tumors. Most of them are using either multimodal images, that means both MRI and WSI, or segmented part of tumor for classification to achieve better performance. However, in this research, we used the whole MRI to provide a more meaningful interpretation of the output. Now let's move into the methodology. We applied interpretability to MRI classifying deep learning model. This is the high-level architecture diagram of the methodology. There are two basic modules where one is for segmentation and the other one is for classification. VAE3D U-Net is used for segmentation while DenseNet is used for classification. Segmentation results are in a semantic segmented MRI volume. Classification results give logics for each of three classes. Then the interpretable approach of the classification model is carried out with respect to the most dominant logic and eventually creates a heat map superimposed on the MRI. This interprets the decision-making process of the classification model. Now we can achieve transparency, which in return gives a human understandable model. This is the segmentation module, which is based on variational autoencoder 3D U-Net, in short VAE3D U-Net. The VAE branch is used only in the training phase to regularize the encoder. DenseNet architecture was used to implement the classifier because of its ability to perform well with a lower number of parameters compared to other architectures in classification tasks. We have used a DenseNet BC model with 169 layers. The original DenseNet BC model was modified to accommodate 3D convolution to accord with 4D inputs. Trained classification module was subjected to breadcrumb process, which will be discussed in the next section. Now we shall look into our contribution, which is the interpretable approach. The contribution of our research is to apply interpretability into deep learning models. So we plan to apply interpretability to the MRI classification module. Through this approach, we can get a visualization output of the contribution level of each region towards the decision-making process of the model. An image is a result of our approach. Dark red colored regions are the regions which are highly contributed towards the decision-making process, while dark blue colored regions are the regions which are slightly contributing towards the decision-making process. Accordingly, this visualization is more human understandable and it resolves the black box nature of deep learning models. To acquire interpretability, we utilized the gradient-weighted class activation mapping, which is in short GradCam. This is a high-level diagram to elaborate how GradCam is functioning. This scenario is a multi-class classification problem. First, we input an image. Then, features are extracted through a convolutional neural network and finally classified using a fully connected layer. Eventually, we will be getting logits for each of the classes. Then, in GradCam process, we find the gradient of the most dominant logit concerning the feature maps of the final convolutional layer. These gradient maps are in equal dimensions as of the feature maps of the final convolutional layer. Next, these gradient maps are averaged channel-wise and multiply the final convolutional layer feature maps with its respective gradient map average value. After that, all the maps are globally averaged to have a single map. Then the result will be scaled up to match the dimensions with the input image. Then this heat map will be superimposed on the input image to make it more human understandable. When compared to other interpretable approaches, the GradCam does not require retraining, means GradCam is applied to train models. Also, it does not require a specific architecture. To implement GradCam, we used the M3D-CAM library. Although GradCam finds the gradients concerning the final convolutional layer feature maps, the generated gradient maps were very small in dimensions in our case. When upscaling the gradient maps to the original dimensions of the input image, we lost many information. Therefore, as a novelty approach, we tried generating gradient maps to different layers of the CNN and found the best matching layer to create them. Let's discuss results now. A segmented tumor can be split into 3 subregions. We have used red to indicate edema region, green for non-enhancing region, and blue for enhancing tumor. The union of all 3 regions is generally named as whole tumor. Tumor core is the region belongs to enhancing and non-enhancing tumor, which corresponds to blue and green regions. This is the segmented output predicted by our segmentation module for one of the test cases compared to its ground truth label. The images shown are slices taken from the middle in coronal, axial, and sagittal views of the 3D MRI. We compared performance of segmentation module of the proposed system with the performance of original VAE 3D U-Net model. There is a 2 to 5% difference in the dice coefficients. Use of a different dataset can be the reason for the slight variation. However, this variation is not a concern because we use segmentation output only as an approximate evaluation of GradCam heat maps. Here you can see balanced accuracy, F1 score, and kappa coefficients corresponding to the confusion matrix of the test set. These values are quite close to existing classification models trained on similar sized datasets using similar resources. Since our primary objective is to apply interpretability, we only need a well-performing system that our interpretations are meaningful. Improving accuracy more than existing solutions is not one of our objectives. Using this image, we can understand the effectiveness of interpretable results generated by the proposed model. Here, each column shows segmented results, GradCam results, and original input MRI image, GradCams superimposed on top of the original image. This is the final output from our proposed model. Color range at the bottom indicates the contributing level of each region, where dark blue for less contributing regions, while dark red for highly contributing regions for classification. When comparing segmented results and GradCam results, we can see tumor core has highly contributed. Rest of the whole tumor has slightly less contributed, and rest of the brain has the minimum contribution for classification. Therefore, we can use segmented results for the same image as an approximate evaluation of GradCam results. Heat maps are generated at 2 different depths for the same test case to interpret how decision-making process of classification model is affected by different regions of input MRI at different depths of the input volume. Unlike the previous image, we can see the tumor is unidentifiable by naked eye at certain depths, but results from both GradCam and segmentation module are identical, confirming correctness of our approach. Finally, we shall move to the conclusion. As a summary of our work, we have used VAE 3D U-Net and DenseNet 3D architectures for segmentation and classification modules respectively. GradCam has been used as to introduce interpretability to classification module. Traditional GradCam approach is tested on all hidden layers of the classification model to select the best layer for generating heat maps. Segmentation results were used to evaluate GradCam results. GradCam heat maps can be used as an alternative for segmentation where pixel-wise accuracy is not required. The scope of the proposed model is not limited to MRI. It can be extended to other imaging modalities as well. Thank you.